Standard biophysical studies often analyze proteins in isolated, dilute water solutions. The living cytoplasm is entirely different. It is a dense, chaotic, gel-like environment, packed with proteins, carbohydrates, and nucleic acids, violently jostling for space. This environment is governed by macromolecular crowding. Macromolecular crowding is the principle that high concentrations of macromolecules occupy a significant fraction of the total volume in a cell. This results in steric volume exclusion, a thermodynamic effect where molecules physically block each other from occupying the same physical space. This creates a critical blind spot in instructional biology. Determining protein stability and folding pathways in a dilute buffer fails to account for the extreme spatial restrictions native to a living cell. Achieving an accurate thermodynamic model of protein folding and stability requires artificially replicating this dense crowding environment in the laboratory. Dr. Sherif Abbas at Middle East Technical University designed an experiment to isolate the variables of physiological crowding. The target of the study was human serum transferrin. Human serum transferrin is an 80 kilodalton iron transport glycoprotein responsible for regulating iron in the blood. The researchers analyzed the protein in both its apo, or iron-free, and holo, or iron-bound, states. To simulate the dense cytoplasm, the team introduced specific crowding agents, 70 kilodalton dextrin and Fickle polymers. In the literature, these large synthetic polymers are widely classified as chemically inert. The primary diagnostic tool for the analysis was Fourier transform infrared, or FTIR, spectroscopy. FTIR spectroscopy is an analytical technique that measures how chemical bonds absorb infrared light, specifically targeting the proteins amide 1 and amide 2 vibrational bands. The experimental stakes rely on the definition of those polymers. If dextrin and Fickle are truly inert crowders, they will merely constrain the protein's physical space without altering its structural chemistry. The baseline structural analysis begins by comparing the dilute and crowded samples of transferrin at an ambient 25 degrees Celsius. This chart shows the deconvoluted amide I band for the samples. The two spectral curves perfectly overlap right at 1654 inverse centimeters, the frequency corresponding to the protein's alpha helices and irregular structures. Macromolecular crowding does not induce any shifts in the static, baseline secondary structure of the protein. The analysis then shifts from static structure to structural dynamics. The researchers tracked hydrogen-deuterium exchange via the amide 2 band, a process that measures how easily heavy water penetrates the folded protein. Plotting unexchanged protons over time, dilute holotransferrin shows a steep 63% exchange rate over 60 minutes. With the dextrin crowder, permeability drops threefold to 21%. Steric pressure forces the protein into a rigid, compact conformation, shielding its core from the heavy water solvent. A crowded environment preserves the protein's native secondary structure, but it severely restrains its dynamic flexibility and solvent permeability. The researchers used thermal perturbation to test thermodynamic stability, gradually heating the samples from 25 to 90 degrees Celsius. In a standard in vitro buffer, extreme heat causes the loss of the protein's alpha helices, followed by the formation of extended intermolecular beta sheet aggregates. In the 1D FTIR spectrum for dilute holotransferrin at 90 degrees, the primary peak shifts leftward from 1656 down to 1647 as the structure denatures. In the crowded sample, the peak barely shifts, but novel aggregation peaks emerge at 1616 and 1638. Volume exclusion restricts the spatial degrees of freedom required for the standard unfolding sequence. When thermodynamic stability finally fails under extreme heat, the protein is forced to adopt completely different aggregated conformations. 1D FTIR captures the start and end states of denaturation, but it cannot resolve overlapping intermediate species or confirm the precise sequence of structural breakdown. Synchronous 2D IR uses temperature to spread overlapping peaks across a second dimension. On this holotransferrin map, negative cross-correlation peaks at 1642 versus 1657 show that the destruction of alpha helices occurs simultaneously with the creation of aggregated beta sheets. Synchronous 2D IR proves a direct, concurrent thermodynamic relationship exists between native structure failure and aggregate formation. Asynchronous 2D IR mapping plots out-of-phase variations. This allows researchers to determine the exact sequential order of structural reorientations, moving beyond just tracking concurrency. Comparing asynchronous 2-dir mats of dextrin and ficol, we see their breakdown sequences do not match. Ficol lacks the specific negative cross-peak at 1674 versus 1659 seen with dextrin, and introduces a strong positive cross-peak at 1640 versus 1624. Because chemically distinct crowders alter the exact sequence and kinetic rate of denaturation, macromolecular crowding cannot be purely a physical phenomenon. Intracellular protein stability relies on two integrated mechanisms: the hard steric boundaries of volume exclusion and the nonspecific chemical interplay defined as soft interactions. The textbook model of volume exclusion, where inert molecules bounce cleanly off a target protein, is incomplete. The cellular interior is a chemically participatory matrix. These transient, soft interactions actively dictate which intermediate folding states a protein can access during high-stress biological events. Future biophysical research modeling aggregation diseases like Alzheimer's must account for specific Crowder chemistry rather than strictly relying on simulated density. The cellular interior is an active, chemically participating environment that strictly governs protein folding. In vivo stability is a completely different thermodynamic landscape than the dilute test tube.